Great. Okay, well, yeah, thanks for joining everyone. Today I'm very pleased to introduce Zonghua Liu, who is from Hong Kong, the Department of Stats and Actuarial Sciences at the University of Hong Kong, who's going to give this seminar on a new MR identification approach, which we're looking forward to hearing. Eric Chechen has joined us as well and might join in the discussion. So thanks very much for agreeing on this. And I'm pleased we worked out that we had a time when Eric would have had to get up at 4 a.m. So we changed it. So and so we've got them both. So I'll leave Lung Hu to start.
Okay. So I want to thank George for the invitation and the kind introduction. So to save time, I will just start. So my talk today is on Mendelian randomization, mixed scale treatment effect, robust identification. So we call it MR mystery for short, and estimation for causal inference. I know the title is a bit long. And this work involves several people, me, Ting from UPenn, Bolo from Singapore, Mary from Hong Kong and New York, Eric from UPenn, US. So the outline of the talk contains 5 parts. First part is introduction, second one is a method, third one is simulation studies. Then we will apply our methods to UK biobank data and then compare with other methods. And in the end, we will have concluding remarks. So the motivation, body mass index and glucose levels. So body mass index, BMI, is defined as the body weight divided by the square of the body height. And glucose levels measures the blood sugar level. The question of interest here is to estimate the causal effect of BMI on the glucose level. Well, in epi studies, we consider several types of designs. We have randomized designs and observational study. Clearly here, we cannot do randomized experiments. To randomize people to have higher or lower BMI is unethical. So we rely on observational study. Well, for observational studies, there are several concerns. For example, confounding bias, reverse causation, selection bias, etc. Especially the confounding bias. If we have unmeasured confounding factors, can we still get unbiased causal effect estimates? You know, in epi study, we try to measure as many confounders as possible. We have huge measurements of covariates, but for a specific question here, the causal effect of BMI on a glucose level, do we have all the confounders? We're not so sure. So if they are unmeasured confounders, can we still get a valid causal effect estimates? Well, that's the question we are trying to address here. So let's go back to the 19th century, the classical Mendelian genetics, especially the law of independent assortment, which says that genes for different traits segregate independently of each other. This is because during meiosis, the chromosomes line up randomly before the cell divides, allowing for gamete formation. Here, I emphasize the word randomly. And the last law is that law of segregation says everyone has 2 versions. We call them alleles for each trait, and one from each parent, one from mom, one from dad, and that these alleles segregate randomly during meiosis. So why talk about Mendelian genetics? Because we need this for Mendelian randomization. Mendelian randomization is an instrumental variable or IV approach that uses genetic variants, for example, single nucleotide polymorphisms or SNPs, to infer the causal effect of a modifiable exposure on a health outcome. So for a SNP to be a valid IV, it must satisfy the following 3 core assumptions. The first one is called IV relevance. The SNP must be associated, well, not necessarily causal association, but be associated with the exposure. It's called IV relevance. Second one is IV independence. the SNP must be independent of any unmeasured confounder of the exposure outcome relationship. The last one, A3, is called exclusion restriction. The SNP cannot have a direct effect on the outcome, not mediated by the exposure. So that means there should be no horizontal pleiotropic effect. Here, figure 2 shows you a so-called a DAG, a directed cyclic graph. This DAG actually encodes the 3 assumptions here. As you can see here, Z is the IV, A is the exposure, Y is the outcome, and U represent the unmeasured confounder. Z has no direct effect on Y. So there's no arrow from Z to Y, and there's no arrow from U to Z. So there's no arrow from Z to Y, and that means exclusion restriction assumption holds. There's no arrow from U to Z, that means the IV independence assumption holds. There's an arrow from Z to A, here, Z to A, that means the IV relevance assumption holds. So this graph, the DAG, encodes the 3 core assumptions. Sorry. Just 1 second. Okay. So even if assumptions A1 to A3 hold for a given IV, a SNP IV, the average causal effect of the treatment on the outcome cannot be point identified. So we need a false condition for identification. There are several assumptions or conditions proposed in the literature. So I will briefly go through them, won't go to the details. So in 1994, Robbins proposed the no current treatment value interaction assumption. Simply says there should be no interaction between the IV and the exposure or the treatment. So here, treatment exposure, they are synonymous, exchangeable. In 1996, Angrist proposed another assumption called a monotonicity assumption about the IV treatment relationship. And they show that the so-called complier average treatment effect can be non-parametrically identified, a so-called case. In 2018, Limbo and Eric established identification of a population average causal effect under the 3 core assumptions and the false condition, so-called no unmeasured common effect modifier of the association between the IV and the endogenous variable and the treatment causal effect on the outcome. The last one is Liu. This Liu is not me, it's someone else. Eric is on the paper. So established identification conditions for the treatment effect among the treated. So effect of treatment on the treated, ETT, on the A1 to A3 and a false condition. The false condition says the selection bias function defined as the odds ratio association between the potential outcome under no treatment, so Y0, potential outcome under no treatment, and the treatment variable is constant across the level of the IV. Actually, we will use this one. As I said, the 3 core assumptions might be violated in practice. Only the first one, the IV relevance assumption, is empirically testable. We can look at the association between the SNP and the exposure. For example, look at the SNP association with the BMI. So this is only one assumption that can be empirically tested. The other 2 assumptions, A2 and A3, we cannot test it. We have data, but we don't know whether those assumptions hold. So violation of A1 is called a weak IV problem. From here, Z to A, if the association between the IV and exposure is weak or null, the A1 assumption is violated. Well, A2 can also be violated, the so-called IV independence assumption. So here, from U to Z, there's a red arrow from U to Z. If this arrow is present, that means assumption A2, the IV independence assumption, is violated. For example, when we have a population stratification, or the selected SNP IV is in linkage, disequilibrium, or LD with another SNP, that SNP has a direct effect on the outcome. If this happens, assumption A2 is violated. So represented by this red arrow from U to Z. A3 assumption, the exclusion restriction assumption, can also be violated. If the SNP IV has a non-null direct effect on the outcome, represented by this blue arrow from Z to Y. The presence of this blue arrow means this SNP IV Z has a direct effect on Y, not through A. This is also called horizontal pleiotropy, which is very common. There's papers showing that this phenomenon is so common in genetics. So that means many SNPs could be invalid IVs. If any violation is valid here, that IV is called invalid IV. So if those assumptions are valid, what can we do? Okay, that's the issues we want to address in this paper, hopefully. So there's some previous work on this topic. The first one is called MRG by E. Assumes that while the candidate SNP may not be a valid IV, but its additive interaction with an observed covariate, this interaction is a valid IV. So they satisfy the three core assumptions. Another approach is called MR-genius. MR-genius relies on an assumption that the residual variance of the first stage regression of the treatment on a SNP is heteroscedastic. Here, we have to be cautious here. The MR-genius is different from our MR-mystery. The heteroscedasticity is about the treatment, the exposure. In our MR-mystery, we will have a different type of heteroscedasticity. A recent paper by Spiller compared this MRG by E and MR-genius and found both methods can be biased when the constant pleiotropy assumption is violated. So both methods can be biased when the constant pleiotropy assumption is violated. So we want to address those issues. Here, I just give you a quick preview of MR mystery. I will talk about the details later, so just give you a quick preview. So we propose an approach called MR mystery that rely on three new assumptions. The first one, the treatment effect, for example, the causal effect, the ETT, does not change with the IV on the additive scale. Here I want to emphasize this is on the additive scale. So causal effect is defined on the additive scale, and then we assume this causal effect does not change across the levels of the IV. The second, the selection bias. The selection bias measures the association between the treatment and the potential outcome under no treatment. So this selection bias due to confounding does not change across levels of the invalid IV on the odds ratio scale. So they're on different scale. So that's the key point here. Causal effect is on the additive scale, bias is on the odds ratio scale. The last, the residual variance of the outcome, not the exposure, the outcome is heteroscedastic and thus must be dependent on the invalid IV. Okay, so we will go to details. Problem setup. Suppose we have independent and identically distributed data. We call it OI. I is the sample index. I from 1 to n, and the sample size. So we have three variables here. ZI is a SNP, AI is exposure, YI is the outcome of interest. Here, YI is a continuous outcome. So for the whole paper, we focus on continuous. So let's use the potential outcome or counterfactual outcome framework to define causal effect. So let's YAZ is YAZ denote the potential outcome. Had possibly contrary to fact, the exposure A and the SNP IV Z being set to little a and little z. And I use this y little a denote the potential outcome had a, the exposure being set to a. Now, we define the causal effect, the effect of treatment on the treated, ETT. This is the parameter of interest in our paper. So this ETT is defined as beta A, which is conditional expectation of YA minus Y0 among A is A. So let's make it simple. Let's consider a binary treatment. So the ETT will be like this. Beta is expectation of Y1 minus Y0 among treated people. So what does this mean? This means among persons who were actually treated. So this causal contrast compares their observed outcomes to that they would have experienced had they not been treated. So this Y0 among the treated people is not observed, right? You are treated. I want to know your outcome if you were not treated. Well, that Y0 is not observed, but this Y1, of course, among the treated, the outcome, if people were treated among the people who are actually treated, is observed by the consistent assumption. So this is really the issue here, Y0 among the treated people. This potential outcome is not observed. So confounding bias, by the consistent assumption, this Y1 among the treated is equal to the observed outcome, potential outcome is equal to the observed outcome among people who are treated by the consistency assumption. But in the absence of confounding, for example, in a randomized study, the treatment or exposure is randomized, in ideal randomized, completely randomized, okay? Then this treatment A is independent of Y0, the potential outcome, which means the individuals who received treatment are on average comparable to those who didn't receive the treatment. Comparable of what? Comparable with respect to pre-treatment prognostic factors for the outcome. This is the purpose of randomization. Like in randomized clinical trials, we randomize the treatment so that the two groups, people who got the treatment, people who don't have the treatment, they're comparable. So here, Y0 is a key prognostic factor. Sometimes people call it the ultimate confounder, because once you know Y0, you don't need the other prognostic factors. So Y0 is a prognostic factor. The independence between A and Y0 in a randomized study, well, that's true, but in observational study, in the presence of confounding, this A and Y0, they are not independent. They are associated. Well, this association is the confounding bias. So in observational studies, the confounding bias is actually captured by the association between this A and this counterfactual outcome Y0. The key observation here is, if we measure this association, well, how to measure the association between two variables, the treatment and the outcome? There's several different scales. You can measure it on additive scale, on multiplicative scale, on other ratio scale. Here, we will measure the association between A and Y0 on the odds ratio scale. This is the key point, one key point here. Now, the difference between the observed outcome Y among treated people minus the average outcome among untreated people, this difference is equal to beta, the causal effect, plus bias. In randomized study, people randomized to get treatment or untreated, this bias is 0 in randomized study. In observational study, when we have a confounding, this bias is non-zero. So our work is trying to tease these two apart, okay? What can we do? In epidemiology, we can do so-called stratification. We stratify the population by the IEV. This IEV could be invalid. So even if we have a SNP IEV that is possibly invalid, we can stratify the population by the value of this SNP IV. So we stratify the whole population by Z. So have this equation conditional on Z value. And we get the difference on the left-hand side is equal to beta Z plus bias Z. Okay, beta Z is the suppose Z has 2 levels, so we have 2 strata. So this beta Z and bias Z, they are stratum specific causal effect and the stratum specific bias. Of course, depends on the value of Z. Now, to understand the identification difficulty, how difficult to identify the causal parameters, let's suppose now, for now, Z is binary. Of course, in general, Z can be anything, but for now, to see the difficulty, assume Z is binary to see how difficult the identification is. Then we have these 2 equations. So this is among Z is 0 stratum, Z is 1 stratum. So we have 2 stratum, and we compare their means, like an average, compare their average. And we have 2 equations here. But on the right-hand side, we have beta 0, bias 0, beta 1, bias 1. So those are stratum specific causal effect and bias. So we have 4 parameters. We have 4 unknowns, but only 2 equations. We all know we have 2 equations, 4 unknowns. We don't have unique solution, right? We have infinite solution, but for identification, we want unique solution. Unique solution means we can uniquely identify beta and the bias. So how to do it, how to proceed? Here is our key strategy here. By imposing assumptions, we can reduce the number of unknowns. We have 4 unknowns, we want to reduce them to 2 by imposing assumptions, so-called homogeneity assumption to be introduced next slide. And we will measure the bias which capture the A and Y0 association on the odds ratio scale. We have to do this on odds ratio scale. If the bias is on the additive scale, the causal effect is also on the additive scale, we cannot tease them apart. Now, here are the 2 assumptions. First one is called a B1 assumption, B2 assumption. First one called homogeneous causal effect assumption, ETT, effect of treatment on the treated. So we assume this causal effect will not change across the levels of the 2 strata, across the level of the Z, the IV. And we also have homogeneous selection bias assumption. So this bias is measured on the odds ratio scale. This is measured by the association between the treatment A and the potential outcome Y0. So this bias does not depend on Z, you can see here. So this gamma parameter, which is log odds ratio, capture the bias. So we have beta, which is the causal effect, bias is the gamma parameter. So assumption 1 essentially says, assumption B1 essentially says that the causal effect of the exposure on the outcome is the same across the levels of the IV. So there's no additive interaction between the exposure and the IV. This is essentially the no current treatment value interaction assumption proposed by Robbins in 1994. And assumption B2 says that the bias does not vary with the levels of the IV. When we measure the assumption, the association between A and Y0 on the odds ratio scale, again, the causal effect is the additive scale, the bias is measured on the odds ratio scale. For example, the causal effect of BMI on glucose level does not change across the level of the SNP IV. And also, we need to assume that the confounding bias is assumed to be balanced across the levels of the SNP IV. That's how to interpret these 2 assumptions. Now, we still need a 3rd assumption. So 2 assumptions is not enough. We need another assumption. So again, in this paper, we focus on continuous outcomes. So let's assume Y, the outcome, is generated according to the following location scale model. So Y is the continuous outcome. It has a mean, conditional mean here, and this is the variance. And we assume this error term epsilon, this error term follows standard normal, and this is a key assumption. But standard normal, normal 0, 1, mean 0, variance is 1, and sigma square z is the variance of y conditional on a and z. So this is a simple location scale model, mean variance. Our primary result is that under those assumptions, the following representation must hold for the observed data. You can see y, the conditional mean of y among A is A, for example, A is treated, conditional on Z is equal to the conditional mean of y among untreated, conditional on the IVZ, plus, this is the causal effect beta, plus bias. And the bias has this form, gamma times the variance of the outcome y conditional on z. So if this sigma square is a constant, that means no outcome heterodicity, we cannot identify beta and gamma. So think about this one is just 1. Then beta plus gamma times A. So you can never tease apart beta and gamma. They go hand in hand, they're together. So to tease beta and gamma apart, this guy, sigma square, conditional on Z, cannot be a constant. It must change in Z. So this motivates the third identification assumption, B3, to identify beta and gamma, the effect, cause effect, and the bias parameter, this sigma square Z cannot be constant. So we need the outcome heteroscedasticity. This is different from homogeneous. Okay, now we have all the 3 assumptions. So with B1, B2, B3, we can identify beta and gamma. So our identifications adventure is here now. So we have all the identification assumptions, 3 assumptions together. Here is a simple graph to show you our identification assumption. So this green line represent conditional mean of y among untreated conditional on z. So you change z from 0 to z to 1, it will change theta z, so this theta z. And then this red dashed line, which is parallel to the green line, represent the hypothetical setting when the outcome variance is a constant. Then what you have is beta plus gamma. You cannot tease them apart. You need heterodicity of the outcome. So if this sigma z is not a constant, that means this b is not 0. Then you can estimate gamma and beta. So we want this solid red line. We don't want this dashed red line. If you have the dashed red line, you cannot identify the causal effect beta. You cannot identify gamma. Okay. And you may ask, is the outcome heterodicity common in genetics? The answer is yes. So there are a lot of evidence there in genetic study to show many quantitative traits, their variance can change across levels of the SNP genotype. Here is an example. So figure A, it shows you the homoscedasticity, and the figure B shows you the heteroscedasticity. You can see here the variance of the mean changes, but at the same time, the quantitative trait, the variance looks like a similar, okay, homoscedasticity. Here, the mean changes, but the variance also changes. The variance gets smaller from genotype 0, 1, and 2, a SNP genotype. Actually, people do GWAS study and found that this particular trait here, this MOT1 gene, if you have C allele here, the quantitative trait, the distribution is this purple one. If you have G allele here, the distribution of the quantitative trait, this green one. As you can see, the variance is changes. The 2 variants are different. If you have C allele, you have smaller variance. If you have G allele, you have larger variance. So the quantitative trait their distribution can be different. Their variance can be different if you have different allele. So this gives you evidence that the quantitative trait, the variance can be different, can be controlled by genes. You may ask, do you have any evidence? Yes, I show you some evidence here. There are actually many papers, including some papers on Nature. I didn't list them, but I give you some evidence. So the variance of a quantitative trait can change across the levels of the SNP genotype. So you may ask, why is that the case? Why the variance can change across the levels of the SNP? Because some gene by independent environment interaction, this interaction can contribute to this heterodicity, outcome heterodicity. Actually, some people already showed that if you have gene by environment interaction, the variance will change across the levels of the SNP IV. I think in 2010, some paper, interesting paper. So gene by environment interaction, sometimes by gene by unmeasured gene environment interaction can contribute to phenotypic variance. Some examples in the literature. Now, here is the statistical model. Suppose we have data and we want to estimate those parameters. We want to build a model. So we build a linear model for the causal effect. Here is the mean, which is beta times A, the exposure, gamma A sigma square, theta 0, theta 1. This is the SNP IV. And we propose a log linear model for the variance. Because the variance is non-negative, so we need this log linear model. So we have these two models. Based on these two models for the observed data, we have this conditional distribution Y given this, which is a normal distribution with this mean and with this variance specified in this model. And we have this normal data for the outcome. What can we do? We propose a novel three-stage estimation procedure, which can give us a consistent estimator, but not fully efficient. And then we propose a one-step update estimator to get a consistent and a fully efficient estimator. Fully efficient simply means the estimator is the most accurate in terms of standard error. It's very accurate. So the confidence interval will be shortest, the standard error will be smallest, a fully efficient estimator. You may ask, why don't we use maximum likelihood estimator, which is simple, you have normal data. Because the likelihood function may not be convex or concave, so there's some numerical reasons we're going to do this. But in the end, this one-step estimator, our estimation procedure still achieve the full efficiency. How to do this? Now, three-step. Run a linear regression, just a simple linear regression, least squares. You regress Y on A, on Z, and A times Z, this interaction term. And then you get residuals, right? You get a residual from linear regression. And then you square the residual, you get a residual square, and you regress on this Z, the IV, in a log linear model. So you get this regression coefficients, eta 0 and eta Z. So this eta Z measures the IV strength here, the association between Z and the outcome variance. So eta Z measures the IV, how strong is the IV. And the third step, you regress this quantity on A and A times this squared. This fitted value from this second regression. Now you are done. You get regression for beta head and a regression for gamma head. You can also do this one for the last step. For the last step, you can regress Y on A times sigma square Z, which is a fitted value from the second step, and Z. You can estimate the beta and the gamma. The estimator will be consistent, even if they're not fully efficient, but they're consistent. And then once we have this three-step estimator, we can use the one-step estimator to update this. Now we have this likelihood, we have this normal distribution, we can write down the likelihood, we have log likelihood, we can calculate the derivative, first order derivative, second order derivative, so we have the score, and then we have the Hessian matrix, and we use this simple iteration. It's kind of like a Newton-Raphson iteration. we only need one step iteration. We get our estimator, the tilde star. So tilde 0 is from the 3-step estimator. We plug in this equation, we get theta tilde star. So tilde star will be consistent and fully efficient. Okay, so let's move on to weak IV. Suppose in your data you have many weak, invalid IV. They are weak, they are also invalid. Can we combine those weak IV to mitigate the weak IV bias? The answer is yes. You simply put Z as a multi-dimensional vector. You just put it into the model, this model. So we have one Z here. You just put many Z here. Okay. Then we have likelihood, and then you can estimate those parameters using the approach called conditional maximum likelihood estimation, CMLE. It turns out under certain regularity conditions, those conditions are very mild, means easy to satisfy. And we show that the estimator is consistent and asymptotic normal. So we can use this to calculate the standard errors, to construct confidence interval, and get p-values. Now, we assume the error term is the standard normal. Can we relax that? Yes, we can relax that. We can assume they follow a key component, K like 2, 3, 4, key component Gaussian mixture with mean 0 and the variance is 1. This assumption is much more general and more flexible than the normal error, because when K is 1, this model reduced to the normal error model. So this model is much more robust and flexible and more general, and can capture a large number of non-Gaussian residuals. So later we'll do simulation to show this model really works more robustly. Here is some simulation results. So the sample size is 100,000 to mimic the UK biobank. Actually, UK biobank sample size is much larger than this. So we have one SNP IV from a Bernoulli distribution with minor allele 0.3, and the treatment is normal distribution from normal 0, 1, standard normal. And the outcome Y is continuous and generated from this model. And it's a normal distribution. Normal distribution means here, variance is here. And we change this eta Z. As I said, the eta Z measures how strong is your IV. So we change it, 0.2, 0.15, 0.1, 0.01. It's getting smaller and smaller. So here, this table shows you the estimation is really good. The bias is small. The standard error estimate is accurate. The confidence interval coverage is correct, or close to the nominal ones. And we can estimate the beta, the causal effect. At the same time, we can also estimate the bias parameter gamma. Bias is okay, controllable, slightly larger. For the gamma estimate, the bias is slightly larger. But our primary interest is beta. So the bias is very small. The coverage is okay. It's very close to nominal, 95%. So if it's zeta Z, the RV strength is small, for example, 0.05. Here, the second last row, 0.05, you have a slight, a little bit larger bias, 1.46%, not so large, but if you increase the sample size, still have this same eta Z, the bias will be reduced to 0.36%. Okay, so you have weak RV, kind of weak. You can increase sample size to compensate for that. We also did simulation to combine multiple, many weak IVs. Here we try 20 SNPs and 50 SNPs. The results show that the coverage is correct. The bias is small, and so it looks very good. We also do simulation on the Gaussian mixture error. So we have tried two components. You have two component Gaussian mixture error for the error term. Here is the setup. Two normal distributions. We have a Bernoulli random variable. We also, if it's 1, we'll take this one error. If it's not, it's 0, we take this one error. So this Gaussian mixture, and then we generate a continuous Y follow this distribution. This is the mean. This is the variance, and the conclusion, the finding is that our coverage is good. And if your eta Z is very, very small, the coverage may not be good. But you can, if you increase your RV strength eta Z here, the coverage is good by the small. Okay, standard error is okay. The coverage is not quite quite good if your eta z is very small. Okay, so it gets better when your eta z is stronger, when the IV is stronger. So we apply our method to the UQ biobank data. The total sample size is here, is roughly 300,000. And to control population stratification, we focus on white British people. And we look at the body mass index and the glucose level. Glucose level is the outcome, BMI is the exposure. And here is the result summarized in this table. So the first 4 columns, beta, standard error, beta, gamma, standard error, gamma, we use normal errors for the MR mystery. Here are the estimates. So beta stars, gamma star, those we use Gaussian mixed errors for MR mystery. You can look at their estimates. So last 2 columns, we use the so-called 2-stage least square, the classical instrument variable method for continuous outcome, 2-stage least squares. You can see the 2-stage least square, the estimate can be very different from MR mystery. So we combine 20 SNPs, 13 of them are kind of weak. And we get this estimate. The causal estimate is 0.08, which is, has a very small standard error. P-value is highly significant. And we also estimate the selection bias, the gamma, which is positive and highly significant. Standard error is small. We also tried MR Genius. So MR Genius, we get this estimate, 0.046, kind of close to our estimate here. But the MR Genius cannot give you a gamma estimate, so we don't report gamma estimate here. And we did a crude regression analysis. This means we simply regress glucose on BMI, just regression. Glucose on BMI, it's called crude regression analysis. This is the causal estimate, which is kind of close to MR Genius, also our estimates, at least in this table. Also highly significant. So we include 7 SNPs into the regression to adjust for that. We get this estimate. So kind of close to this crude estimate. And to take this SNP, this particular SNP as an example. So our estimate, MR mystery, give you this for normal errors. And selection bias this, okay? And if we use the standard 2-state least squares, they give you negative estimate. Of course, the p-value is not significant. So you can see the two-stage least square can be very different for this SNP. This SNP is likely to be an invalid IV, okay? An invalid IV. That's why the two-stage least square can give biased results. To conclude, MR mystery leverage the association between the SNP IV and the variance of the outcome to disentangle confounding bias from causal effect of interest, which is the outcome heterodicity assumption. And gene by unmeasured independent environmental interactions can contribute to the outcome heterodicity, which is fundamentally important for identification in our paper. And we can combine many weak IVs, invalid IVs, to reduce weak IV bias. So in the future, actually, we have some results already. So the error term for the continuous outcome can be further relaxed to be totally unrestricted. We don't have any parametric distribution for the error term using our semi-parametric approach. And we are going to work on the robustness and efficiency of this semi-parametric approach. Currently, we are also developing methods for binary outcomes. We have made some progress. Hopefully in the future, in the near future, we can share our results for binary outcomes to you. Here are some selected references, and our paper has been posted on medRxiv for a while, I think in September, late September. Here are the co-authors. I want to thank all my co-authors, Ting from UPenn, Paulo from Singapore, NUS, and my colleague Mary Schooling from Hong Kong U, also CUNY, and Eric from UPenn. want to thank our sponsors. My research is sponsored by Hong Kong U. Eric research is sponsored by NIH. We are the grant numbers. And Bolo's research is sponsored by NUS Singapore. This is a grant number. And we also thank UK Biobank. This is our application number. And thank you all for your time and listening. And I think I have presented all I have to you. Thank you very much.
Thanks very much. That was a fantastic seminar. Raises lots of questions, but really written very clearly presented. If people would like to ask questions, if you raise your hand in the participant section, then Marie will unmute you, and then you can ask questions. Or as I say, you can type them in the Q&A place. There's now just a thanks to Eric for answering the questions by in there. So I'll make just a comment on a question about this. So the comment is with respect to what you say about the actual presence of difference in variance by genotype. Now there have been several studies, and I'll post them in the chat section. I'll post the links in the chat section. Yeah, exactly. There've been several studies which have now used this approach on a genome-wide scale with every SNP, a huge number of phenotypes in UK Biobank, and this is by Peter Fisher's group have done it in one way. Peter Donnelly's group have done it in the other way. They've found identical results, which is that there's a very low prevalence of robustly identified variance differences, because of course you're testing so many genotypes against so many phenotypes that in fact they get 78 significant for every genotype against every continuous phenotype in UK Biobank. They get 78 identified cases where there is robust evidence of variance difference. I mean, the really nice thing about that is the top one is what many people would have guessed would have been the top one. It's a variant related to smoking heavily and lung function. And of course you get variance differences because it only has an effect in the people who smoke, and the people who don't smoke, it doesn't have an effect. So you get a 10 to the minus 10 heterogeneity as a statistic on variance differences. So my question is to include SNPs. I'll post all those references in the chat. But to include the SNPs to say that they do show variance difference, what level of evidence are you taking? What's, you know, I mean, you took 7 SNPs which you thought were heteroscedastic. How did you include those? I mean, what degree of, seeing as you could test so many against, you could examine the variance differences,
yes,
a lot of SNPs. What level of evidence did you take? And do you think there's, because of course there would be overfitting problems if there'll be chance fluctuations, of course, in any particular study will lead to evidence of such. And then if you're applying that in the same study, you would have overfitting issues.
Okay, so thank George for the helpful comments. Here we have 20 SNPs here because we look at another paper that use UK Biobank. They look at the exposure there in that paper. I think it's by Sun. The paper look at the BMI and the blood pressure. BMI is exposure. They have a list of SNPs that are associated with body mass index, not a variance level, but mean. And we simply look at their SNPs. So that's why the multiple testing burden is much more smaller. We don't need like a 5 times 10 to the minus 8 the level to choose the significant SNPs. So we only have like 20 SNPs. We look at how many passed the significant level in our second stage regression here. So
that's using a p is greater than less than 0.05 level, is it?
We use a 0.05 divided by 20. So still accounts for multiple testing, but not like millions, but we only have 20.
Yeah.
So this is how we choose the SNPs based on our previous research. They provide a list. We look at the list. They are a kind of a BMI SNP IVs. We just look at them. So I agree with that. If we do a genome-wide scan to look at the SNPs that are kind of related to the variance of the trait, the multiple testing burden is huge. Probably we don't have, cannot find many, probably 10 or 20, I don't know, depends on the sample size. So the power issue is also another issue. But luckily, UK Biobank, we have a huge sample size. So probably we can identify many SNPs that are associated with the variance of the trait.
I also wanted to say that that's a really good point, George, because we are really concerned with these types of methods about weak IV problems. And the weak IV problem here manifests itself if, in fact, what you said is the setting you're in, that many of the SNPs might only be weakly associated with your phenotype defining the outcome. In which case, you really do have to think about weak IV bias. And that's part of the reasons why in this paper we also showed how one might be able to address that issue, because the concerns about weak IV are much stronger here than they might be in other settings where all you need is an association on the mean scale. And so that is the other consideration we gave to this issue with this work.
So is there any would there be any equivalent to a sort of F test for your instrument strength for this?
Zhonghua,
yeah.
Okay, so did you? Okay, so you can use similar statistics for looking at the weak instrument.
Yeah, okay. Similar is similar to F test, yes.
Yeah. So there's a question in the Q&A from Chin Yang saying, what happens if some instruments don't satisfy the heterogeneity assumption?
What if well, if you're asking one SNP or all of the SNPs? If no SNP satisfy, we cannot identify, right? If no SNP satisfy the heterogeneity assumption, then we cannot identify, which is a key identification assumption for our approach.
And fortunately, this is an assumption that is again testable. It's exactly the point that George was raising. In principle, because we're living in this world where we're using linear models as a way of gauging what the causal effect might be, if you only need one or a subset of your Z's to satisfy the assumption in order to identify this causal effect. And so even if you were in a situation where most of them did not, but a few of them did and had a strong enough association on the variance scale, then you would again recover identification.
Okay, thank you. That makes sense.
Wes, do you want to ask your, if Marie can unmute Wes, then Wes can ask a question.
There we go. So yeah, thanks for like a really clear and informative talk. I was just curious with using these, using weak IVs, whether there was sort of a point of diminishing returns or sort of a cutoff, because generally when you're incorporating more and more weak instruments, sometimes there's a temptation to throw as much as possible in. And I wasn't sure if maybe there was a point where you would limit the weak instruments you were including.
That's a very good question. Eric, do you have any comments on that?
Yeah, so if you look at most of the literature on weak IV, and this is very well developed in so valid but weak IVs in the econometrics literature, it's very well developed literature. The usual arguments is in an asymptotic or large sample arguments where the sample size grows, the number of IVs also grow. However, they cannot grow too fast relative to sample size. So this is addressing your question as to how do you scale the number of weak IVs relative to the amount of signal you have and the amount of information you have in your study. And so there's a careful balance between you can't just take everything and then throw the whole genome in and say it doesn't matter if it's not associated, you're gonna get killed. And so we do have, I mean, it's a little bit of a technical answer, but there are some in the paper we give conditions related to the last bullet point here as to how many weak IVs can you consider for a given sample size, and it cannot be too large. And it kind of makes sense because at the end of the day, you do have to run regressions on all of these IVs. And so if you have more candidate IVs or the number of IVs that scales with the sample size, you're just not going to be able to incorporate them. So there are formulas that give you exactly that scaling factor, which we can't really get into it now, but we'll be happy to further discuss this. And it's all tied to the information matrix of which we call I here, and how that might scale with the dimension of the number of IVs.
Thank you so much.
Great. And I don't know if there's any. Are there any other questions, Marie? If there are, if you're on mute, someone. So we haven't got another question. I'll just make a comment which Eric said before. So, but which is that in the way this is a mirror image, sort of doing the reverse of approaches basically introduced by R.A. Fisher in 1938 for using differences in variance by your randomized group, saying that the difference in a difference in variance in the treated versus untreated group suggests the presence that there are interactions, that globally you should see a variance change of the treatment if there is any interaction, and that then you'd need to think that there's sort of heterogeneity in response, but that if there are, and this has been now used in randomized controlled trials to say, that well, it looks as though the treatment is having a similar effect in the whole group, and there's not any important sized heterogeneity, any important group which is getting a different effect, so therefore, by analogy with with Fisher's suggestion, you could say that a variant which is allowing you to estimate the population average effect will be one where the variance is not different. So if you have the same variance across the genotypic level, which is your instrument, that would say that you would have, unless some completely implausible perfect balancing effects are going on, which are implausible, then that allows you to estimate an effect. It doesn't tell you that your instruments are valid, of course, because you could perfectly well have a genetic variant which had just a homogeneous effect on your outcome that is invalid. But it means if the instruments are valid, then you are getting a treatment that you are getting a population average treatment effect out of your estimate. So that approach would suggest you want to make that estimate. You exclude the you exclude precisely those variants that you're including to allow a population average treatment effect estimate.
That's exactly right. And if I may add, I mean I couldn't say it anymore. That's exactly right. If I might just add that in addition to that, in addition to having to select your SNPs such that the treatment effect might not interact with those SNPs, there is a second requirement here, which is where the name comes from, this mixed identification strategy, that in addition to that, the amount of selection bias has to be the same across levels of your SNPs.
Just SNPs. Yeah. Okay.
And so it's even more than what you're saying. And all of this goes to the fact that as we know, you need some pretty strong assumptions to identify a causal effect in these situations where you don't have a valid IV, and this is simply articulating what they might be, how you might reason about whether or not you're in a situation where you could recover such a causal effect.
Yeah. I think Emma wants to be unmuted. Oh, okay. No, Emma doesn't want to be unmuted. The method, of course, well, it seems depends on you having a continuous outcome, or at least an outcome you can put into some form which allows you to have some sensible variance estimate. I mean, you couldn't use it with a one-naught outcome, could you?
Zhonghua, do you want to?
Could you apply this method? You applied it to an example, obviously, with glucose as the outcome.
Yes.
I mean, is there any way of applying it to a categorical outcome? Heart attack, no heart attack, because you don't have a variance for categorical outcome.
I think for categorical outcome, we need a new methodology for that. Is that right, Eric?
Yeah. Because so we Zhonghua did mention that we are working on extensions of this work to binary outcomes. That was the last point, and this would also include categorical outcomes. The real key to any of these approaches is not necessarily the heteroscedastic error really comes into play only in a situation where you have a continuous outcome. When you have a binary outcome, for instance, then just because the variance of a binary outcome, it depends on the mean, the usual, if you have any association between your candidate SNP and the outcome at all, that would imply heteroscedastic outcome, and so that assumption almost always holds. What remains important in those settings is that the scale on which you're measuring the causal effect and the scale on which you're measuring the bias due to measure confounding do not coincide, and so that remains the key, the core assumption in those settings. However, there's some technical difficulties, which is why we're still working on this, because as we know, binary outcomes must satisfy some boundedness conditions, like the mean has to be between 0 and 1. And so to ensure that all of these conditions actually are coherent, to ensure that there are some data distribution where they all mutually hold, actually requires a very careful analysis of how you might parameterize the likelihood for such data sets. And so this is ongoing work, but we're pretty confident. I think we've worked out most of the theory, and we're pretty confident that it would go through.
That's great. Sorry, I now realize I've run over the time, even though Zhonghua left lots of time for discussion. I'd like to thank the speakers. I mean, it's terrible in these seminars, you get no clapping. We should have some sort of clapping simulator or something. It was a really great seminar, and I think a few of us will join on another link for continue a bit of discussion in a few minutes. I think Zhonghua and Eric, you should have had another Zoom link to go on to.
Yeah, no, we do.
Okay.
Thank you so much.
Thanks again. I'll be on there in about 3 minutes.
Okay.
Bye.
So we have a link.